Before I continue, let me quickly show you that these ones you see here, these are the bases present in each of them. So these are the bases, these are the bases. This one is not fully shown. Now this part is the sugar phosphate backbone. So this strand, sugar phosphate backbone, this ones are the sugar phosphate backbone. So these are the bases. Now let me quickly mention that the bases in DNA, we have thymine, we have adenine, we have cytosine, and we have guanine. So the same way in RNA, we have adenine, we have cytosine, we have guanine. I get it. But the difference is just that instead of thymine in DNA, we have uracil in RNA. So both of them contains 4 bases altogether, joined together. So now let me quickly tell something that in DNA, take note, thymine is always joined to adenine in DNA. Thymine always joined to adenine, and the same way, likewise, cytosine is always joined to guanine. Are you getting it? That's in DNA. So now, they pair together. That's how it occurs. So, but in RNA, the pairs we have the same way. We have cytosine and guanine. They pair. So now, don't forget that I said in DNA, thymine and adenine join together. Are you getting it? So in RNA, it is uracil and adenine that joins together. Why? Because in RNA doesn't have thymine. So that's just the basis of that. The basis of that. So those are the bases. Now the bases are joined by hydrogen bond. So the bases, they are joined by hydrogen bond. So let's move forward. We talk about the K. So let's talk about complementary base pairing in DNA. Now there's what we call complementary base pairing. So complementary base pairing is, is a process in which thymine is made to bond to adenine, in which the bases are able to bond with themselves. So I mentioned earlier that thymine always bond to adenine and cytosine to guanine. So we are discussing DNA now. So now, how is this possible? How do we achieve complementary base pairing? Complementary base pairing is achieved by making, I mentioned earlier that DNA is made up of 2 helical strands. Two helical strands. So now this helical strand, now for complementary base pairing to occur, they must run opposite of themselves. What are we saying when we say they must run opposite of themselves? It means that the 5' of one helical strand is started first and ended with 3' and with 3', and the other helical strand starts with 3' and end with 5'. So that's the idea there. Now when we talk of the 3' and 5', what are we really talking about? The phosphate groups in DNA, the phosphate groups in nucleotide, they are joined to the carbon 5 and the carbon 3. So let me show you that now. Now look at what we have now. This is a 5 carbon sugar. Now this is a 5 carbon sugar. That's this is a, okay. Don't forget in DNA we have deoxyribose. So this OH here, this H here, this H here. Now if it has been ribose sugar, we would have had OH here, but because it's deoxyribose, so we have H here. So the same thing goes on and on. Then we have HO, we have CH2 here. So now this is carbon 1, 2, as I've shown you earlier. So now the phosphate group. Here, one phosphate group is joined here. Can you see? This one phosphate group. Here we have another phosphate group. So can you see? So now this carbon 1, this carbon 3, this 1, 2, 3, this carbon 5. Are you getting it? So now the same way also if another one, so this is another sugar here, this is another sugar here. So we have OH. Okay, sorry, this is a, okay, let's put the base here. Let's assume that the base here, this our base here is adenine. Let's represent it with A. So the same way, so now our base here, another base here, let's put cytosine. So now look at here, this is the fifth one, CH2. So now also bonded here, are you getting it?